thank you very much for joining us today, Liam. And yeah, I hand over to you.
Yeah, thank you very much. Yeah, thank you for having me. Very excited to talk about the recent work that we've been doing. So yeah, as was mentioned, I am a postdoctoral researcher at Northeastern University in Boston with the Spring Lab. And so we have recently been delving into this ablation area where we haven't really been doing any sort of ablation before. A lot of our work is largely focused on imaging or different treatment modalities, something like PT, if any of you are familiar with that. But so this ablation process is sort of very new for our group, and we had to sort of learn a lot along the way as we were doing all of this work. So today I'm just going to be talking about specifically this nonlinear ablation and how the pulse duration impacts your ability to perform this ablation at high efficiency and without sort of destroying anything that you don't want to actually destroy with this powerful laser. And so to start, I just want to make sure everyone's on the same page when I'm talking about this these nonlinear optical processes, because this is really sort of the crux of what we're doing here. And so in general, these here you see this image of just a single photon fluorescence versus this 2-photon fluorescence, and this really sort of showcases the main aspect of this nonlinear ablation that we're trying to take advantage of. And so we want to be able to really heavily restrict the target volume that we're performing the ablation in, similarly to how 2-photon fluorescence imaging heavily restricts the fluorescent volume that you're actually imaging. And so in order to utilize all of our expertise, we have this pretty large team of collaborators to do this precise ablation. So we have several people in the Spring Lab in Boston, but then we have a couple of collaborators over at Cornell University, Henry and Frank, as well as Omir Eldai over in Germany at Ruhr University, who is a ablation expert. And then Pablo Valdez, who is a neurosurgeon at the University of Texas Medical Branch, who is sort of guiding our medical decisions as far as what is safe and what's not for these ablation processes. And so why do we care so much about this precise and efficient ablation to begin with? Well, largely it's to do with this idea of high-grade glioblastoma treatments. And so there's been quite a few advancements in sort of how patients are treated for these this sort of really severe disease. But even with sort of the advancements of medical tools and the actual surgical process itself, such as this fluorescence guided surgery, which you can see here on the right, the actual overall survival for patients that receive this diagnosis really hasn't improved all that much, as you might expect, given the huge amount of advancements in the actual applications of our technologies, et cetera. And so one reason for this is this idea that there's just almost always this recurrence of the disease, regardless of kind of how well the surgeon can kind of get at the tumor. And this comes around to the idea of this complete resection. And so, as you can imagine, if you're performing surgery in the brain, you have to be extremely careful as far as what tissue to remove and not remove. So for example, if you have, say, cancer in your liver, they can just cut out a massive chunk of your liver to make sure they definitely get all of the cancer, and you'll be mostly okay. But in the brain, obviously, you can't just cut out a huge chunk of the brain and make sure you get all the cancer. So there's a lot of protocols in place to sort of prevent you from going too far into this sort of sensitive tissue. And so because of this, speaking with like several neurosurgeons and neuropathologists, the issue is that they're knowingly leaving behind cancer when doing these surgical procedures. They can only remove so much safely. And so we want to be able to give neurosurgeons the ability to then go after sort of these microscopic deposits that they're knowingly leaving behind with the control and safety that they would require to actually go further into the marginal areas around the main tumor and actually remove these microscopic tumor deposits to hopefully avoid any sort of recurrence that they often see in these marginal regions. And so how are we planning on doing that? Well, this is sort of building off of some technology that we've already been working on, but our idea is that we want to be able to use an existing device in our lab to scan for these tumors, these microscopic tumor deposits, and then just ablate them safely and remove them from the surgical bed. And so this is largely building on some work from previous members of the Spring Lab. So Kai Zhang, he developed this small handheld microendoscope that is capable of really nice video rate fluorescent imaging. And so we have done quite a bit of work to actually be able to find these cancer cells that are fluorescently tagged. But now a lot of what we are working on is how do we remove them safely and avoid sort of damaging the healthy brain tissue around those tumorous deposits. And so how are we planning on doing that? Well, Omir Eldai, as I mentioned, over at RUR, is a ablation expert, and he developed this idea of ablation cooling. And so in this method of ablation, what you have is this sort of manipulation of the laser pulse train, where you take all of the laser pulses and you tightly pack them into what are essentially called laser bursts. And due to that tight packing of these laser pulses, you get this really efficient ablation process within that laser burst. And you see that the tissue is effectively erupted off of whatever sample you're ablating. And it is happening so quickly and so efficiently that the heat really doesn't have time to dissipate out around where you're ablating to the quote unquote healthy areas. And so this really serves to restrict the actual damaged volume that you're ablating. And and just as a note, if any of you have any questions, feel free to just jump in and say so. I'm happy to answer as we go. But as you can imagine, this ablation process that Omir developed is quite technical and complicated. And this sort of came as I was rounding down my PhD. So I was sort of faced with the decision of, do I start an entirely new PhD when I'm about to finish my first one? Or do we sort of work on sort of a stepping stone towards this that can sort of mimic some of the beneficial capabilities of this without needing to completely restart all the work that we've been doing. And so just to sort of set the stage as far as what people typically do for ablation, I just want to quickly talk about ablation regimes and sort of what they typically look like and how they're typically used. And so usually any sort of ablation falls into either this low repetition rate regime or high repetition rate regime. So you either have these very high energy pulses that are coming in at something under usually a megahertz range. And so these high energy pulses can trigger ablation. With each individual pulse, you're forming plasma as you actually send these pulses in. It's a very destructive but effective method for ablation. And then you have this high repetition rate regime where you have very high laser repetition rates up above 100 megahertz with very low individual pulse energies. And so you see this sort of cumulative effect of the ablation where each individual pulse can't necessarily trigger the ablation, but as more and more pulses come in, this cumulative effect of this energy deposition starts to ablate away the material in the region. And so as was mentioned, Brian actually gave a talk here a little while back about our laser that we had, which is a 32 megahertz repetition rate. And so we wanted to utilize what we had already. And so instead, we're actually looking at how we can perform this ablation in this intermediate regime where we hopefully can take advantage of higher pulse energies than you might see in this high repetition rate regime, but with a much higher repetition rate than you see in the low rate regime, and sort of try and meld the two and hopefully generate something that's sort of the best of both worlds in this scenario. And so in order to do this, we have an existing Andy laser, which the laser pulse energies were just not sufficient for actually performing ablation. And so we needed a way to amplify these laser pulses to generate high enough laser power to actually perform this ablation. So we're using something called gain managed nonlinear amplification. And so this is a method of laser amplification developed by the WISE group over at Cornell. And so for any of you that might be less familiar with laser amplification, a really common method for doing so is called CPA. So essentially you will take your laser pulse, you'll stretch it out using some diffraction gratings, and then you'll take that stretched pulse, send it through a gain fiber, and as it travels through, it's amplified in that gain fiber. And then at the output, you can recompress your pulse so you have a very powerful and very short pulse at the output. And so the difference here is for GMA is that you send in a very short laser pulse into your gain fiber. And as it travels through the fiber, as you can see in this video here on the right, the pulse actually is broadened. The spectrum of the pulse is broadened as it travels through the fiber. So you're getting that stretching effect, and it is also amplifying as it travels through the fiber. And so at the output, you just need a pair of gratings to recompress this stretched pulse. And you're left with very high energy, very broad bandwidth pulses with very short pulse durations. And so traditionally with CPA, you can get on the order of 100 femtoseconds. And this is sort of without any higher order dispersion accommodations that you might need if you want to go even shorter with CPA. But with GMA, you can just use 2 grating compressors and get extremely short pulse durations, which can be really beneficial in particular if you're looking to maybe use this sort of system for imaging. So you can get pulses similar to, say, if any of you are familiar with like SLAM microscopy, which is done by Steve Beaupart. You can get these really short pulse durations that allow you to actually look at endogenous fluorophores within the body, or even second harmonic generation, third harmonic generation, even if you're really, really careful with how you set things up. So it's a really simple and effective method for amplifying and generating these really clean, short pulses. And so we, as I mentioned, we have this the 32 megahertz Andy laser that Brian actually spoke about when he was here before. So we're using that laser as the seed for our new amplifying setup. And so in this blue box here is the actual amplifier portion of our system. And so Henry from Cornell was nice enough to visit and help us set up this system. It's not overly complicated as far as what components you need, but the setup can be a little bit less scientific and more of an art sometimes as far as getting everything really nicely aligned with all of the separate collimators and gain fibers and which pump settings you need, etc. So his help was really beneficial to getting this set up in a much quicker way. And so the idea is that you take the seed pulse from the anti-laser and you truncate it using this very narrow bandpass filter so that it is as close to transform limited as you can get. And then you send it in through a collimator into this 10 micron core gain fiber, this ytterbium doped gain fiber. And you pump that gain fiber with varying different powers from this pump laser in order until you sort of fall on which sort of output you want. And then at the output, you can get this really broad spectrum pulse. So we get these pulses that are approximately 150 nanometers in bandwidth. And after compression, we are able to see pulses that are sub 50 femtoseconds down towards 40 femtoseconds even, depending on how cleanly we set things up. And so we use this amplifying system for our ablation experiments. We just send the output to a pair of Galvo scanning mirrors through a couple tube lenses to direct it, and then through this simple aspherical lens at first anyway, and then to the sample. And so we align this sample to the edge, which I will explain why later. But we just use a simple camera, CMOS camera, to actually watch the ablation, and then we just raster scan along the surface of the sample so that we can generate some ablation grooves in the sample and actually then look at sort of how the pulse duration affects this and how well we're able to actually ablate and how quickly. And so this is an example of sort of the experiments that we're running with the doing some ablation on silicon wafers. And so you can see sort of this pulse duration on the right. So we alter the pulse duration and look at how that affects our ability to generate these grooves in the silicon surface. And then on the left is just a video. It might be a little bit hard to see with the frame rate, I'm not sure. But essentially you just, we position the laser towards the edge and then we do a quick raster scan off the edge of the silicon. And so the reason for this being at the edge of the silicon is that we really initially struggled with actually measuring the depth and width of these grooves that we're generating. It was particularly difficult just due to they're quite small and narrow, so it was particularly difficult. And so what we settled on was actually turning them on their side so we could look at the grooves shape. And so just really quickly before I get into that, I'll just talk about sort of what we wanted to look at as far as how this depended on the pulse duration itself. And so the reason we started with silicon is just that it's really rigid and it has a somewhat similar absorption ratio to that of tissue, at least in the wavelength that we're working on around like this, centered around this 1080 nanometers. And also really importantly is that it's got this pretty well-established ablation threshold that is documented in literature quite a bit. So it's a very easy starting test bed for these initial ablation experiments when we have had very little experience with ablation prior to this. And so what we did was we started with this, the Gaussian intensity profile of the laser, and we broke that up into the axial and the radial components. So just at the center and then along the axial direction. And so looking at these two components broken up now, where we're either at the or axially or radially, and just because it's easier to measure the power of the laser directly rather than the intensity, we make this quick substitution where we can look at this equation in terms of the laser power and this Rayleigh length substitution as well, just to make everything have the same set of variables. We can come to this sort of equation where we can compare the threshold intensity where you see ablation and compare that to the actual radius or depth that you can achieve with a set power. So that's this ITH is just this well-established intensity threshold, and then the ZTH is how deep will the ablation be if we have some set power, and then this rho is just how wide will that groove be if we have some set power. And so then we can rearrange things and rewrite things in terms of the pulse duration tau. And so we get these two variables, this zth and this rho th, and using those as the two axes of an ellipse, we can determine a model for how large these ablation grooves should be based on the average power that we're using and our repetition rate and pulse duration that we're using, etc. And so now here is sort of where I was talking about this silicon ablation and turning it on its edge so that we can actually look at the grooves, because from above in B, this is what you see when you look at the ablation grooves in the silicon. It's a lot harder to tell sort of the actual depth and width of these grooves with traditional imaging methods. And so we just turn it on its side so we can look at the orthogonal face of the silicon and see these grooves from the side. And then just using reflectance microscopy, we can actually look at these grooves. And then just to show you what these two variables are specifically, so the depth of these grooves and then the radius of these grooves are the Z and rho respectively. And then in A here, you can see some examples, admittedly blurry examples, I apologize. They're quite small, of these different grooves that we generated with different pulse durations. So you can see here in I, for example, this is the shortest pulse duration that we ended up using, which was approximately 44 femtoseconds in duration, up to this much longer pulse duration where we're about at 2 picoseconds here. So there's quite a obvious difference already just visually. But when we plot these along with the fit that we generated with the rho and Z, we see this pretty good agreement with the theoretical model that we were assuming would be correct. But you see this huge improvement just from going from this hundreds of femtoseconds pulse durations down to this 40 or so femtosecond pulse duration. And that's without adjusting the laser power or anything like that. It's purely just adjusting the actual duration of the pulse itself. So you can maintain a lower pulse duration and achieve a much higher ablation efficiency just purely by playing around with that pulse duration. And so in this blue circle here, you can see sort of this usual range where you can get an off-the-shelf version of a ablation laser. This is sort of where those pulse durations will fall for one of those off-the-shelf models. And so using this custom setup, we can actually get a much shorter pulse duration and therefore a much more efficient ablation process to occur. And so this is a great result to begin with. We're really happy with where we've started here anyway, but obviously we're much more interested in sort of how this works in biological media as opposed to just a silicon wafer. And so we tried initially doing some tissue ablation, and if you are looking at these images and not sure what you're looking at, that would make a lot of sense because we also didn't really know how to tell how well our ablation was working based on this. So these are two confocal microscopy images of chicken breast that we ablated with our laser. And as you can see, it's really burnt and very heavily distorted, really difficult to tell what's going on. So instead of doing this tissue ablation directly, what we decided was instead to look at a more simple model, which was cellular ablation or cell microsurgery. And so in order to do this, there's quite a few differences between the silicon setup and the Cella setup. But the main difference is just that in the silicon setup, we had a very simple, just a aspherical lens, which was used for actually focusing the beam. But in the cell setup, we have a much fancier microscope objective for ensuring a really high NA, which ends up being a very important feature of the ablation at such a small scale so that we can restrict the actual laser ablation to a much smaller region without sort of destroying everything around it. And so I can come back to these parameters if anyone's interested in them, but the main idea is just that we replaced our initial aspherical lens with this much fancier laser microscope objective so that we could really improve the NA and focus of the laser itself. So this is the setup that we were using for a large portion of our cellular ablation experiments. And so you can see again, the galvo mirror here. We have some two lenses for expanding the beam, and then just the microscope objective and a simple Thorlabs camera to actually see what we're doing in real time. And so these are some of the videos that we have of the experiments. So you can see on the right here, you have this pulse duration. And then as you do the experiment, you can see these sort of what I call grill marks start to form on the cells as you're actually scanning the laser across the cell surface. And so this is the basic version of the experiment that we ran. So we have these fluorescent cell line, this fluorescent cell line with GFP inside of it. So we have these OVCAR 5 GFP cells. We cultured them in plates, and then we performed these raster scans of the laser across the cell surface. And then we put PI dye into these plates, so propydium iodide, which is a very common live dead stain for cells. So the way that it works is that it generally will stay outside the cell until a cell is dead and the membrane of it is permeable. And so by ablating across these membranes of these cells, we're opening up the membrane and allowing the PI dye to flow into those cells. And so when it does, it becomes a very bright orange fluorescence under our laser. And so we can see how many of these cells are actually killed under these different scanning parameters and sort of get an idea of how effectively we're actually permeabilizing these cells. And so what that looks like under fluorescence microscopy is something like this over here on the right. So we have in B, we have this example where we have the really short pulse duration, this 50 femtosecond pulse duration, and we just did 2 raster scans across the cell membrane surface, and you can see quite a few of these cells have uptaken this orange PI dye within the scan region, which is denoted by this red rectangle here. And so it's very obvious, very bright. And then in C, we did the same process where we're raster scanning the laser across the cell surface. But now we're using 175 femtosecond pulse duration. But in order to generate even this sort of lackluster amount of propydium iodide uptake, we had to perform 15 scans just to get something even similar to these 2 scans that we performed at 50 femtoseconds. So it's a pretty stark difference just in that small difference of pulse duration in our ability to actually permeabilize these cells to the propydium iodide. And then in D is just a control no ablation example, in case you were wondering. And so, yeah, so we have all of these different pulse durations on a variety of different, a multitude of different plates, and just see something very similar to before where we see a much improved intensity of the PI dye in the scan region per scan for the pulse duration that we use. So as we go shorter and shorter, we see a much brighter fluorescent signal from the PI dye per scan that we actually had to perform across these cell membranes. And so due to the complexities that are involved with the biological system like this, we didn't go into a full in-depth theoretical derivation of sort of how this is happening, because then you start to get into diffusion and other mobility processes of this actual dye flowing into these cells. And that was sort of beyond what we wanted to look at. So instead, we just we fit to just a inverse square dependency of this pulse duration, and it fits quite well. So there's clearly this inverse square relation between the permeabilization of these cells and the pulse duration of the ablation laser that we used. And so all of these cell membrane experiments were done in 2D culture, just a flat plate. But we wanted to obviously sort of look at how we could potentially transition this to something more relevant to the broader perspective initial start of this project, which is the surgical use of it. So we started to look at how well we could actually ablate within 3D culture. And so what this is here is we take 3D matrigel domes. So it's just a commercial product. It's an extracellular matrix that you can buy that mimics just the extracellular matrix of a normal tumor environment. And then the OVCAR5 cancer cells will grow within that dome in these sort of spheroids all over the 3D space. And so we can then take that dome that we have and perform ablation at varying depths within that dome and look at how well we can actually ablate several hundred microns down into that dome. So we can look at how well we can control the ablation effect at depth and how deep we can actually go within a medium like that. And so here we have just a 3D rendering of this dome when we did perform the ablation. So you can see a few of these spheroids we performed the ablation on. And then as an example, this is sort of just a top-down view of what one of those spheroids looks like following the ablation. And so this was really, we were really happy with this, but we wanted to, given that we're sort of doing this with the overall idea of being able to do a surgical process with something like this, we wanted to look at how well controlled this process was. So we looked at the planar scans of these spheroids and fit the intensity of the PI dye again to just the square of this intensity profile of the beam itself. So again, just looping back to sort of this square dependency of the actual process, we wanted to look at the square of the dependency and fit that to this in order to look at how effectively we're actually controlling this Z depth restriction of the ablation. And so what we did here is that you can see in A and in C, there are 2 peaks to this fit, and that's because due to the depth that we're at, we actually performed the ablation scans at 2 separate planes within the spheroid. And so that you can see that captured in the model here. At this depth, you're getting this sort of distortion of the beam as it travels through the scattering medium. And as the actual cells start to get scanned across multiple times, you start to heat up the cells and you get this sort of expansion of what should be approximately 7 microns as far as the actual focal restriction of this. And it sort of expands to this like 11 microns in this case due to the distortion of the medium as well as the actual local heating starting to cause off-target killing. So it's definitely distorted and expanded beyond sort of the ideal case, but it still falls quite close to the overall ideal restrictive capabilities of this objective. So we think that even though it's expanded somewhat, it's still within the range where it's considered controllable and precise enough where we can avoid damaging huge portions of tissue outside of where we're actually trying to look at in particular. And so this was sort of the final sort of good news that we were looking for when performing these experiments. And so if you want to look more in depth at sort of what we did with this, I would highly encourage you to take a look at the paper that we just published regarding all of this. But that is sort of what we've been doing with the ablation, and we're sort of moving towards some more ablation experiments in the future where we're looking at the effect of fluorophores on those experiments and how those contribute to the actual efficiency of the ablation process, et cetera. But that is sort of the scope of what I have been working on recently. And I just want to thank the members of the Spring Lab and Brian, who, as mentioned before, gave a previous talk about our base laser oscillator, which I would highly recommend you all take a look at if you're interested in sort of where the starting point of the system is. But yeah, if anyone has any questions or anything like that, I'm happy to answer them.